week 4 of your biology class. The topic for today's class is nucleic acid, which is a very important biomolecule in all living cells. We know that organic molecules are carbohydrates, proteins, lipids, and nucleic acid. We have so far already learned about carbohydrates, proteins, and lipids. Now the fourth organic molecule is nucleic acids. We have all heard that our genetic information is transferred from generation to generation, or we acquire some genes from our parents that we transfer to our offsprings. This is the important role of nucleic acids in the body. It has also another important function, which is protein synthesis. Now let's look at the learning objectives. I have divided the lesson into 3 separate sections. In the first section, you will learn about the basic structure of nucleic acid, which is the nucleotides. Then you will study the 2 examples, which is DNA and RNA. After section 1, you will pause the video and take a short quiz. In section 2, you will study about the structure of DNA and RNA. After section 2, you will pause the video again to take a short drawing assignment. Finally, in section 3, you will learn about the differences of RNA and DNA and its functions. Before going to section 1, I want you all to recollect some information that we learned from our previous lessons. Like when we learned about the carbohydrates, we learned that carbohydrates was made up of smaller monomer units called monosaccharides, which form the larger polymer polysaccharides. When we learned about proteins, we learned that it was made up of smaller monomer units called amino acids, which forms the bigger polymer polypeptides or proteins. Lipids were made up of fatty acids and glycerol. Similarly, nucleic acids are made up of the monomer units called nucleotides, which forms the bigger polymer or polynucleotide, also known as nucleic acids. 2 examples are DNA and RNA. If you look at the basic structure of nucleic acid, it is made up of smaller monomer units called nucleotide. That is, monomer nucleotide are built up to form a bigger polymer, which is nucleic acid, or it is also known as polynucleotide. Now we look into the basic structure of one of the single monomer unit, a nucleotide. Or what are the components in this nucleotide? It has 3 basic components. A phosphate group, which is attached to a pentose sugar. The pentose sugar is attached to a nitrogenous base. All these 3 units together make the nucleotide. When the pentose sugar is just attached to the nitrogenous base, it is called the nucleoside. But here our focus is on the nucleotide. Now we look into each of these 3 components one by one. Like what are the nitrogenous bases made of? Or which pentose sugar are we talking about? First, the nitrogenous bases. The 2 types of nitrogenous bases that are seen in nucleic acids are purines and pyrimidines. The purines are adenine and guanine, abbreviated as A and G. Pyrimidines are cytosine, thymine, and uracil. Thymine is usually seen in DNA, and in RNA, the thymine is replaced by uracil. If you look at the chemical structure of purines and pyrimidines, you can see that pyrimidines are single ring structures and purines are double ring structures. You can also see the nitrogen attached to the rings. Now the second component of the nucleotide monomer, that is the pentose sugar. There are 2 types, 2-deoxyribose and the ribose. It is a pentose sugar because it has 5 carbon atoms. Always remember the carbon atoms are numbered in a clockwise direction. If you look at the second carbon atom in the ribose sugar, you will see that there is an OH group or a hydroxyl group. This oxygen is missing in 2-deoxyribose. Remember that in DNA, the pentose sugar is always 2-deoxyribose because the oxygen is missing. While in RNA, it is always ribose because the OH group is present. Now the third component, the phosphate group. The structure is very simple with a phosphate in the middle and 4 oxygens surrounding it. And that is why we usually represent the phosphate in the form of a circle. Now if we go back to the nucleotide structure, it has a phosphate group which is attached to the pentose sugar. The pentose sugar are of two types, deoxyribose in DNA and ribose in RNA. Pentose sugar is attached to the nitrogenous bases. The nitrogenous bases are adenine, guanine, cytosine and thymine in DNA, and in RNA, the thymine is replaced by uracil. The two examples of nucleic acids are DNA and RNA. But when we studied about the energy carriers of cell in mitochondria, we learned about another nucleotide structure, ATP or adenosine triphosphate. From the name, we'll understand the nitrogenous base here is adenine attached to the ribose sugar, and there are 3 phosphate group. But today we'll study about DNA or deoxyribonucleic acid and RNA or ribonucleic acid. We have come to the end of section 1. Now I want you to pause the video and answer these 4 questions. Feedback to the questions. The nucleotide structure has phosphate group pentose and nitrogenous base. The purine nitrogenous bases are adenine and guanine. The 3 pyrimidine nitrogenous bases are thymine, cytosine and uracil. And the difference between the nitrogenous bases in DNA and RNA is that thymine in DNA is replaced by uracil in RNA. If you have got all your answers correct, proceed to the second section of the lesson. If you have got any answers wrong, I suggest you replay the video and answer them again. Continue until you get all your answers correct. Now proceed to section 2 of the lesson. Now the structure of DNA. You see that there are 2 polynucleotide chains or 2 polymers. Therefore, DNA is called the double helix structure. You can see that there are 4 monomer units on the left hand side and 4 monomer units on the right hand side. If you look at the phosphate group, it is attached to the 5th carbon atom of the deoxyribose. While on the right hand side, the deoxyribose has been inverted. It's because the nitrogenous bases always attach to the 1st carbon atom of the deoxyribose. You see the thymine has been attached to the 1st carbon atom. Therefore, here the phosphate is attached to the 3rd carbon atom of the deoxyribose. This gives the spiral structure of the DNA. This also forms the antiparallel nature of the polynucleotide chains, that is 5' to 3' on one side and 3' to 5' on the other side. The deoxyribose is attached to the phosphate by phosphodiester bonds. This forms the backbone of the DNA. The nitrogenous bases in the middle are attached by hydrogen bonds. The nitrogenous bases are always complementary to each other. That is, purines always base pairs with pyrimidines. That is, adenine base pairs with thymine by 2 hydrogen bonds, and guanine always base pairs with cytosine by 3 hydrogen bonds. This is a simple structure of the DNA. You can see the sugar phosphate backbone and the nitrogenous bases in the middle. The nitrogenous bases are adenine base pairing with thymine and guanine base pairing with cytosine. Adenine 2 hydrogen bonds base pair with thymine. Guanine 3 hydrogen bonds base pair with cytosine. You can also see the antiparallel nature of the 2 polynucleotide chains. 5' to 3' in one direction and 3' to 5' in another direction. This is the most simple representation of the DNA structure, the spiral staircase model or the double helix structure. The deoxyribose is attached to the phosphate group by phosphodiester bonds, and this forms the sugar phosphate backbone of the DNA. The antiparallel nature of the DNA is also very important, especially when you learn about DNA replication in future. The nitrogenous bases in the middle are attached by hydrogen bonds, and this gives the DNA a more stable structure. Adenine always pairs with thymine and guanine always pairs with cytosine. Now we move on to the structure of RNA. You see that it is a single polynucleotide chain. Here the thymine has been replaced by uracil. The pentose sugar here is ribose. You see the hydroxyl group attached to the 2' carbon. Sometimes RNA itself coils to form some complementary base pairing. In that case, the adenine pairs with uracil with 2 hydrogen bonds and cytosine pairs with guanine with 3 hydrogen bonds. Now this is the simple representation of RNA. Unlike DNA, you see that it is a single helix or there is only a single polynucleotide structure. The sugar ribose is linked to the phosphate group by phosphodiester bonds, forming the sugar phosphate backbone. You can see that in the brown. Another thing to remember about the RNA structure is about the nitrogenous bases. The thymine has been replaced by uracil. So the nitrogenous bases are adenine, cytosine, guanine, and uracil. Now we have come to the end of section 2 of the lesson. Now pause the video and try to draw the structure of DNA and RNA. Check your drawing by going back to the video if needed. Now we come to the final section of the lesson, that is the differences of DNA and RNA. Remember that DNA is a double helix structure because it has 2 polynucleotide chains linked by hydrogen bonds in the middle, while RNA it is a single helical structure. The pentose sugar in DNA is deoxyribose because the oxygen is missing from the second carbon atom, while in RNA it is a ribose sugar because the oxygen or the hydroxyl group is present in the second carbon atom. The nitrogenous bases are adenine, guanine, cytosine, and thymine in DNA, while in RNA the thymine has been replaced by uracil. DNA stores the genetic information in the organism, while RNA's function is to transfer this genetic information from the DNA to the ribosome for the synthesis of protein. Now the functions of DNA and RNA. DNA is a storehouse of genetic information of the cell. That is, each segment of DNA, say the gene, code for a particular genetic information. For example, say the eye color, or genetic information for the synthesis of proteins like collagen, keratin, etc. DNA is present in the nucleus of the eukaryotic cells. So who helps to transfer this information to the ribosomes, which is present in the cytoplasm? This is done by 3 types of RNA: messenger RNA, tRNA, and rRNA. So these RNA helps in the transfer of genetic information from the DNA in the nucleus to the ribosome in the cytoplasm for the synthesis of proteins. So both the DNA and RNA helps in the synthesis of proteins. So our next lesson will be on the synthesis of proteins. We will learn 2 processes: transcription, which is the genetic information transferred to the messenger RNA from DNA, and then translation, which is the synthesis of protein in the ribosome by the information brought in from the messenger RNA. The assignment for this week, you have to discuss in your teams and answer the following questions. What are the 3 major differences of DNA and RNA? And the 2 main functions of DNA and RNA. Thank you for watching.